Good afternoon. The title of my project is Microbial Communities Associated with Soil Health, which is a pretty broad subject area. Initially, the idea was to support the ENOS project to help the team understand the changes in microbial community composition and activity. At the time that my project was proposed, there was a companion proposed project to look at volatile organic compounds, but that didn't receive funding. So I decided to take this on board as well, as this is something that was critical for the ENOS team and something that I'm interested in also. So for my first experiment, I decided to look at the volatile organic compounds that were being exuded from the soil and whether they could be used to detect changes in microbial activity. To be sure that I was looking at microbially produced VOCs, I needed to eliminate the other potential sources of VOCs. I proceeded to address this in the form of a pot trial with no macrofauna or plants present. I believe that the best way to affect microbial activity in the absence of other living organisms was to stress the communities to look at what effect this had on VOC emissions. This picture here is just one of my pots with a prototype ENOS. Each of the pots in my pot trial had its own ENOS that remained in place for the duration of the experiment. For the pot trial, I used two types of soil that were commonly used here in Tasmania, a ferrosol and a sandy loam. We decided to use the plant available moisture range to keep the temperature constant. Tensiometers were used to determine these points. Water stressing was chosen as the preferred method of stressing. It was hypothesised that emissions would be lower when the soil was saturated and the microbes that contribute to plant health, i.e. soil health, were unhappy, and that the emissions would increase as the soil dried out and the microbes became happier, until the soil became too dry and emissions would once again decrease. We hypothesised that the emissions would look something like this. But did they? So these graphs are just a sample of what we found. Looking at a range of alcohols emitted from ferrosol, the graphs on the left, and the sandy loam on the right. When looking at the alcohol group of emissions, we did see what we somewhat expected. Although there were other patterns emerging, such as where the highest emissions were recorded at the saturation point, as well as variations between the ferrosol and the sandy loam. It starts to get a bit less clear cut when looking at the other volatile groups, such as the aldehydes, where we see the greatest production at different moisture points, primarily at the permanent wilting point, as well as seeing some more interesting patterns dependent upon the compound. Now, statistical analyses confirmed that the composition of bacterial communities showed strong differences between soil types, so we took a closer look at some of the key genera. These graphs show, for example, relative abundances of a few genera from the order Acidimicrobiales. This is an order of Actinomycetes, which are of great importance in agricultural and forest soils. You can see that most of these increase in relative abundance until the soil dries to permanent wilting point before declining. This contrasts with the typical pattern seen in these genera from the family Acidobacteriaceae, which contains many plant growth promoting species. Several genera in Acidobacteriaceae increase in relative abundance at the very, very dry stage. So, as there are so many species in genera in these samples, we have to start to consider interactions among species and whether some of the VOCs emitted are dependent on interactions between the species. This is where the fun begins, as well as much further analysis. So, how does this affect or help farmers? We've seen that the varying moisture levels affected both the abundance of the microbial communities as well as the VOCs emitted. My second experiment was based in 2 apple orchards and focused on the effects of various mulch treatments on microbial biomass carbon, community composition, and soil VOCs. Now, there's insufficient time to go into the details of these results, but we can see from the graph on the left that that treatments affected the soil moisture, and this was correlated with an increase in microbial biomass carbon, which is an indicator of microbial community size, as seen in the graph on the right. Analysis of principal coordinates has allowed us to identify the bacterial and fungal species that are increased under the treatments, so future work can examine the potential contribution to plant health and VOC emissions, and this analysis is ongoing. Now, I feel pretty confident in saying that most of you, if not all in the room, would appreciate the complexities associated with trying to understand 2 very complex matrices, being soil and microbial communities. and that putting this understanding to a practical, everyday use for landowners to utilise could take a great deal of more time and research. However, with all areas of research, you must begin somewhere, and I believe that this work lays the foundation for very practical future outcomes. Thank you very much.